take a measurement of something that is quantitative, you often get one of these graphs. So this is a histogram again, sorry. Let me just write that out for us. Called a histogram. So if you take measurements and you take a lot of them, you might find that you have lots of measurements close to the, I'll say average. And of course, as we usually say average, we usually mean the mean. A lot of times your measurements are pretty close to the average. So how tall is this plant? Well, it has an average height for the species, and some individuals are a little bit bigger than that, a little bit smaller than the average. And some individuals, just very few of them, are a lot different from the average. So if you take all these numbers and you ask the question on the y-axis of how many measurements you got in each of these categories, you take sort of a sliding range, you'll see you get the most measurements around the mean. You get a smaller number of measurements farther away from the mean. Sometimes it drops off considerably as you go away. So not many measurements far from the mean, lots of measurements near the mean, and you end up with one of these bell curves or normal distributions. And if you love biology, this is something you will find really a lot of different kinds of measurements will give you this same distribution. So whether you're measuring the height of people, skin color, if you could put a number to something, you'll get an average and then lots of variation around that average. So just hopefully you're familiar with this fairly, at least on the surface, familiar with this idea. So there's a mean and then these so-called standard deviations, which are distances from the mean where most of the measurements fall. So most of the measurements are within 2 standard deviations of the mean. So whatever you're measuring, if it's consistent for the species, say you're going for leaf length, then most of your measurements will fall within some range around some specific mean. And the ability to measure that mean accurately is due a little bit to the number of samples you measure. So I just did a simulation here where I was simulating a normal distribution. And if you take a few measurements, sometimes you get one of these more choppy distributions. So you can kind of guess maybe there's about an average around 0. But this peak over at 1.2 is pretty big. It's the biggest one next to this other one. The biggest one isn't at 0. So it's sort of choppy distribution, kind of broadly, maybe following that. This is measuring 100 individuals. So I just wanted to illustrate with this that you get better graphs by measuring more. This is another feature of statistics and biology. So count 100, count 1,000, and then over here 10,000. If you manage to count 10,000 measurements, you get one of these really crisp measurements, normal distributions that follow the pattern. So hopefully if you're able to do some kind of measurements like this, you have a lot of samples to work with. So we might have the experience of measuring a small number, and we might not have the best perception of the mean, but we can maybe rely on that. If we kept doing that, we'd get closer to one of these nice clean distributions. So fortunately, tons and tons of data out there if you ever wanted to find someone else's normal distributions. Here's some examples of different kinds of sorghum. And these are different cultivated versions of sorghum and basically how long it takes them to start flowering. And each one of these, this is called a violin plot. So it's like a normal distribution on its side. Again, the fattest part is usually pretty close to the mean. And the average overall, the average flowering time goes up on the axis. So they've kind of sorted them by that. And you get a lot of confidence in the quality of your average if you measure however many they measured on here. You can also have traits that vary together. So you might have a plant that in good growing conditions makes lots of flowers and lots of leaves, makes big leaves and lots of leaves or something like that. You can take these measurements to go together. That can go along with variation. It could also go with the genetics of telling different species apart. So these are from something I had a lot of data for. On the left, this is a feature that doesn't quite have the same predictive power. They're not super well correlated. The line connecting them, you see it doesn't really seem to follow all the points. And it's also kind of flat. So one of these flat lines means that you're not really associating these two features together. And this was a group of species, a group of individuals, and their blade length and pedicel length. And that wasn't so well correlated together. This is a vegetative feature and a reproductive feature. But over here, the calyx length and the pedicel length, those were a little bit better correlated. So we have a nice upslope on the axis there. And a lot of these points are fairly close to the line. I don't know, take my word for it. So on the right-hand side, these are more correlated or co-vary. Right, correlated on there. If you have traits that are correlated, again, maybe not falling exactly on that line. Sometimes that happens and might be due to genetics or environment. And going back, we already talked about variation within an individual. And what do we expect to find varying within an individual? A lot of times these little finer differences. So fine differences in leaf size, flower number, and so on. This author here, Chitwood, has done a lot of work to investigate variation and basically to see a lot of investigation into plants in the grapevine family, kind of seeing what parts of that variation are consistent. So you can have lots of different kinds of leaves produced by a species, and he and his co-authors are looking for these parts of the measurements that are consistently different or consistently something you could identify the species with. And part of that is this process, which is called ontogenetic change or ontogeny. Ontogeny refers to basically change during development, and plants are different from animals. Animals, we often have this progression toward a reproductive individuals. You get the same thing in a plant, but maybe, I don't know, it's more spaced out over space and not necessarily change within an existing organ. So within a plant, these are the leaves that are produced. We're in the passion flowers now. These are the leaves that are produced during the course of development. So the first leaves are entire, they're small, they get bigger, and then they start getting smaller again, and they start getting lobed. And this might be a transition into the reproductive phase of this plant. So a little bit different from animals in that we're making different organs. We're making new organs, and then they still change over time. Similar thing here, entire leaves, lobed leaves, and then skinny lobes as we go through time, and so on and so on and so on. So lots of variation, important to account for if you're trying to identify one of these species. So in an early phase, you might not be able to tell them apart by their leaves because they're all very similar, but then they turn into these much different leaves over time. So when we talk about ontogeny, this is something that is tied with the genetics of development. A little bit different from environmental. If it was environmental, that's what we talked about before with phenotypic plasticity. All right, and then within a species, lots of differences here. I think I've done a lot of this by now, but just a nice illustration here. Plants grown, same species, grown in the same environment, still producing lots of differences, potentially due to their genetics. So what do we expect to vary? Again, some of these minor differences, sizes, number, maybe within a range that's still diagnostic to a species, and other features that are more constant, maybe some of these bigger differences, or really this is sometimes something you have to investigate. Okay, what is it? I've got these different species I need to tell apart. What is consistently different about them? This is the result of a lot of morphometric investigation. Whew, there's a lot to go on here. My goodness.